Hello everyone. Welcome to today's live broadcast, Advanced Analysis of Complex Cell Models with the new Operetta CLS High Content Analysis System. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Their innovative detection, imaging, informatics, and service capabilities, combined with deep market knowledge and expertise, help customers gain greater insights into their science. Before we begin, just a couple of important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of your presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speakers. We have two speakers today, Jacob Tesdorpf and Joe Trask. Speaking first today will be Dr. Jacob Tesdorpf. As Portfolio Director for High Content Instruments, Dr. Tesdorpf leads global product management and application development for high content screening at PerkinElmer. He holds a doctorate in biochemistry from Hanover University and did research on nuclear hormone receptors at the Max Planck Institute. Dr. Tesdorpf has held several imaging-related sales and marketing roles across proteomics, microarrays, preclinical imaging, and high-content analysis, and joined PerkinElmer in 2010. I will now hand over to Dr. Tesdorpf for his presentation. Dr. Tesdorpf?
Thank you, Christina. Thank you, Christina. Good afternoon, good morning, everybody. I would like to present you our new Operetta CLS High Content Analysis System. And I would like to start this presentation with a little introduction as to why we would want to do high content screening. Drug development is still one of the most expensive efforts in our industry, costing up to 10 years worth of research and $2.6 billion to bring a drug to market. Only 12% of all candidates that enter phase 1 pass this critical clinical phase, and this adds to the cost of drug development. How can we improve that? How can we as PerkinElmer help you to improve this process? Recently, a paper was published by Jack Scannell and Jim Bosley, where they positioned that quality can beat quantity. And they did a mathematical modeling of the drug discovery process to understand how the quality of the models you're using for your research impact your chances of finding good drug candidates. This graph from this paper basically shows the relationship between the correlation of your model predictive value and the throughput that you achieve and your chances of success in this process. What this graph tells you is that even small changes in the value of the correlation between your model and the clinical approval improve greatly the chances of finding good drugs. How can we help you to improve the predictivity of your model system? This is really what the development of high content screening in PerkinElmer is about. Can we help you? I hope yes. One thing that has happened over time was really the improvement of model systems used for high content screening, used for drug discovery. Many years back, people were using simple cell lines like HeLa, and we're looking for simple effects like translocations or simple stains as shown here in this little image. In the past couple of years, a large number of much more powerful and much more physiologically relevant models have come up. Some examples are shown here. This is an example of using primary red ganglion cells. This is an example of stem cell co-cultures, and this is iPSC-derived cardiomyocytes, all used as model systems for drug discovery and primary research. They are much closer to the situation in the human body than the HeLa cells ever were. Increasingly, we see even higher value models coming up. We're seeing more and more people working with live cells. We're seeing model organisms like zebrafish gaining in importance. We see a tremendous increase in working with spheroids and organoids, which are three-dimensional model systems providing a much richer environment for the cells to differentiate in ways that resemble the human body. And we see also an increased work with tissue slides. We see also more advanced models using, for example, FRET to look at protein-protein interaction. All these new model systems mean that you need more imaging power. Once working with primary cells, the phenotype you're looking for becomes more scarce. When working with co-cultures, only part of the cells will be the cells of interest in your image. When working with spheroids, you will want to sample throughout multiple planes to get a good representation of the total spheroid. All these means that the demands for a high-content instrument are ever increasing, both in terms of throughput, image quality, and analysis capabilities. And this is where we try to help with our new Operetta CLS. PerkinElmer has been involved in high-content screening for over a decade now. The first instrument we brought to market was the then leading Opera high-content screening system. It was the first multi-camera laser-based spinning disc confocal system, and at that time had a really innovative, powerful a cappella image analysis software. But that software was an expert tool. It was not most intuitive, but took quite a lot of learning time to get acquainted with it. We took the experience with the Opera and we brought the Operetta high-content analysis system, which was a LAMP-based, simple desktop unit, which combined confocal imaging with a really easy-to-use Harmony software suite, which was hiding the power of a cappella underneath an easier user interface. We developed this software further so that it could deal with more complex assays, and we succeeded the old Opera with the Opera Phoenix in 2014. Here we brought together a laser-based microlens confocal system that could hold 1 to 4 cameras, but being operated by really easy-to-use software. And now, in 2016, we're bringing to you the Operetta CLS. Here we combine the fast mechanics from the Opera Phoenix with an LED-based single-camera system, providing an extremely powerful imaging device aimed at the middle range of the market. At the same time, we have always increased our image analysis and data analysis capabilities, which are inherently important for high-content screening, with Columbus and the TIBCO Spotfire HC Profiler. So, Operetta CLS. We see it as a truly unique combination of technologies, which enables you to do sensitive imaging, to run your everyday analysis every day the right way, to ultimately reveal deep biological insights. What I'm going to talk to you about now is a lot of the technical detail of this instrument, and then in the second part of this webinar, we will hear more of the actual application use of these features. When we talk about imaging, it's all about managing light, bringing light to the sample, and even more importantly, collecting light from the sample and delivering it in a very efficient way to the detector. When we designed the Operetta CLS, we looked at every single step of this process and tried to improve the performance of the old Operetta all the way. During this talk, I'm going to talk a little bit more about the light source, which we now use a high power LED source, which is directly coupled into the light path. We're going to talk a little bit about the spinning disk confocality and what that delivers to the type of applications we're talking about. And we're talking about the objective lenses, which play a crucial role both in delivering light and collecting light from the sample. We also have an 8-position dichroic wheel, which allows you to combine the excitation wavelengths and select the emission wavelengths, ideally matched to every single LED. We have a matching emission filter wheel, which is user accessible so that you can choose the right filters for your assays. And we have a large format state-of-the-art CMOS camera that captures large, high dynamic range, low noise images. In the past, we stayed with lamp excitation for quite a while because we still felt it gave you very good power and very good excitation efficiency. But LEDs have developed very rapidly over the last couple of years, and we felt now that there was a good time to combine the best LEDs we could find into one very compact, very high power internal LED light source. This allows us to provide light with direct injection without any fibers to the sample and switch those between different light wavelengths quickly without slowing down the process. It also means that we have the very same path length for all wavelengths, and it makes it a very stable illumination source. You can choose from a 4 LED set or an LED set depending on the number of fluorophores and the type of fluorophores you want to use. So this light source is internal to the instrument and supports a broad range of fluorophores. We need a lot of light. We need a high excitation energy to also enable true multi-point confocal scanning. The benefit of confocal scanning is clearly to reduce background noise and background blurring. It also improves the resolution in X, Y, and Z, as you can see on these two images. The left-hand image is a wide field image acquired on Operetta CLS. The right-hand image is the confocal image that matches the sample. And if you look carefully, you can see the improved resolution both on the actin fibers and also on the antibody stain here. What you will understand is that introducing a confocal disc reduces the amount of excitation light dramatically. Therefore, we have to have a lot of excitation light entering the light path, and this is what we achieve with our new light source. More importantly is how do we collect light from the sample? And here we introduce for the first time in a system in this performance range water immersion objectives. This is a proprietary technology to PerkinElmer, which we have first introduced in the Opera, which allows us to operate water immersion objectives in true screening conditions. And to visualize the effect of a water immersion objective, we took the same image with the same exposure setting, same exposure time, same exposure power, and also the same contrast settings for display, one time with a very nice 40x 0.7 NA air lens, and now we took the very same image with the corresponding water immersion objective. As you will see, this image is much brighter, actually 4 times much brighter, and it also provides better resolution. This is very beautiful, and it also gives you a glimpse of what is possible with high content screening today in terms of resolution and cellular detail, but it might not be quantitative enough for your taste. The next slide, we try to put a more quantitative view on how different objectives on the Operetta CLS compare. In this experiment, we took a 60-plane confocal Z stack of a stacked HapG2 culture. So if you look on the left-hand side, you will see one plane out of this stack, and you will see up here the XZ view, and to the left you will see the stack in YZ view. What you can see immediately, and this stack has been acquired with a 40x long working distance objective with an NA of 0.6, that the image is somewhat blurry and that it is clearly distorted in Z direction. It took us 34 seconds to acquire the stack of 60 planes. We had to excite at very high power for a significant amount of time, and when we compared one plane before and after acquiring the stack, actually the signal was 27% lower, so a significant amount of bleaching had occurred. When we move to a higher NA objective, we can reduce the excitation energy, so time, exposure time times power setting, significantly to maybe 30% of what we used before. We will reduce the bleaching, and the time to acquire the stack becomes significantly shorter. So this is possible with an air objective. If we move to the very right of this slide, you will see the result as it looks when acquired with a water immersion objective. Please note that if you watch the XZ view here and the YZ view here, that nuclei are much better resolved in Z when compared to either the high NA or the long working distance objective. Also, you can see that the degree of confocality is much better, and much more of the higher plane blur has been rejected from the image. More importantly, though, exposure times could be further reduced, so we're only providing 1/10 of the energy. The stack acquisition is more than 4 times faster than with the long working distance objective, and bleaching has been reduced to minimum. Now you might say this is just an Hoechst image and might be a rather boring example of that, but bleaching of Hoechst is a really good proxy to understand what excitation power might do to your live cell samples. The less bleaching you have in an experiment like this, the better live cells will tolerate long-term extended time-lapse imaging. So managing excitation light is a key consideration when planning for long-term live cell experiments. So this should give you an idea how much water immersion objectives can speed up your research and can improve your research results. This is pretty much what I wanted to talk to you about regarding the hardware of the Operetta CLS. We have a very fast system that will capture up to 100,000 wells in a single color assay or up to 50,000 wells in a 4-color assay during a single day, running extremely accurate and extremely fast mechanics throughout the whole process. Having very powerful excitation light sources, having very efficient light collection optics, and providing a lot of images in a very short amount of time. All of this controlled by a very easy to use software which we call Harmony. I'm a strong believer that usability of high content devices is key in achieving good usability and good use out of your investment. This is why we continuously invest in improving Harmony to provide you access to all the power of the hardware and to enable advanced experiments. With Operetta CLS, we bring a new version of Harmony, Harmony 4.5, and we're going to showcase a few of the most advanced features here. For those of you who don't know Harmony, this gives you a brief idea of how Harmony looks. It's a workflow-oriented interface. It starts with setting up your experiment, running your experiment, setting up your image analysis, and finally looking at the evaluation. For example, EC50 curves. You always set up your experiment or your analysis on the left-hand side. You see your results at a glance at the lower bottom end, and you navigate through images and stacks on the right side of the window. Now, let's discuss a few of the newest features in Harmony 4.5. Most importantly, a plugin that we dubbed PreciScan, which provides you with access to intelligent image acquisition. So what is that? Very typically these days, you encounter some sort of rare event screening where the phenotype of interest is few and far in between inside a given well. Imaging the whole well at high resolution becomes impractical because it would create a huge amount of data in the search of very few cells that are really of interest. The same holds true, for example, for microtissues, which may be located anywhere in the well and where you want to acquire a high-resolution stack. So the idea of PreciScan is that in a given well, you do a low magnification pre-scan. Here, for example, at 5x, you use online image analysis to identify the cells of interest, and then you provide those positions back to the software for a subsequent rescan at a higher magnification of your choice, for example, at 63x. Due to the high accuracy of our stage and the automatic image alignment we can do in our software, this process is extremely precise, allowing you to go from 5x to 63x without missing a cell, basically. I'm going to show you this on an example we've done on a Situ slide, which you see here. Situ provides micro-patterned plates, micro-patterned slides, and this is one example. These slides are covered with a pattern which only allows cells to adhere to these little triangular shapes that you can see here on the left, and typically only one cell will adhere. But what you will do during seeding, you will seed a certain number of cells, and some of these patterns will be covered by no cell, some patterns will be covered by one cell, and some few patterns will be covered by two cells. But to really benefit from this patterning, all you're interested in are those patterns that are actually covered by a single cell. So PreciScan allows you to do a pre-scan at 5x, here on the left-hand side, run an image analysis using all the building blocks in Harmony to identify exactly those patterns that contain one cell and only one cell, and then direct the rescan at 63x to capture, for example, a stack at those positions. And as you will see on the next slide, this works actually quite nicely. So as a result of the PreciScan routine, what you get in this case are 6 image stacks, and here you see a maximum intensity projection of 6 patterns, each pattern occupied by a single cell. And what you will also see is that the positioning is extremely precise. So this is a really useful tool which opens up a whole range of new applications to high content screening on Operetta CLS. Another maybe more relevant example to many of you is shown here. This is a plate provided to us by Spherolds, which holds 3D inside microtissues in a gravity trap plate. And as you can see on the left-hand side, this is a pre-scan at 10x. You can see that the microtissue can be found in all different wells and in very different locations. If all you wanted to do is a high-resolution scan of the microtissue, this would become very impractical. So we use this pre-scan, we run an image analysis, and then we do a 20x rescan in this case. and the result as a montage window across the whole plate is shown here. And what you will note is left-hand well was an empty well in the pre-scan, and no image was acquired in the rescan. Maybe also interesting because you were able to configure the image analysis in a very detailed way. Artifacts like here are not recognized as tissue and therefore result in no image in the rescan. Where the microtissue was nicely centered, like here, you can basically see that it is very much centered in the 20x image as well. And this would have worked also for a 40x rescan or a 60x rescan. So because you have access to all the image analysis tools in Harmony, you can tailor the image analysis that you do on the pre-scan very accurately to only pick up the cells of interest. Another tool that we're introducing with Harmony 4.5 is the global image. This is actually a stitching routine that allows you to stitch and downsample the images acquired at high resolution to create a lower resolution overview of your well, for example, as displayed here. So this is a long object imaged at 5x. This is the same object imaged at 10x. And this is the same object imaged with lots of fields at 20x. And we can now basically assemble these high resolution images to give us a lower resolution image to really appreciate the large structure. We use this, for example, to analyze a stretch wound assay. The data was kindly provided by Rune Linding's lab in the University of Copenhagen, Denmark. And what you can see here is the downsampled stitched image, which allows you to detect the wound as shown in pink on the right-hand side here, and which is basically created from the 25 high-resolution images indicated in the lower left-hand corner. Now, why would you want to do this? Is you could do the top left-hand corner image easily with a 5x magnification, but then you would not be able to analyze cells for subtle phenotypic changes, whereas in the 20x image, you would be able to pick up very subtle phenotypic changes, and you can then use Harmony to basically classify phenotypes in relation to their position to, for example, the wound boundary. This is a very useful tool for a wide variety of different applications. Wound healing scratch assays being shown here, but you could also use this to characterize stem cell colonies, 3D microtissues, for example, to define what is the core and what is the boundary of the microtissues, or maybe even for model organisms like zebrafish. With this, I would like to end my part of the presentation, providing you with a final overview of the Operetta CLS, which has one camera, 48 high-power LED, provides you with spinning disk confocality, has a user-accessible 8-position emission filter wheel, and if you look at a 4-color assay, is good for more than 50,000 wells a day. Compared to the Opera Phoenix, which features up to 4 cameras, is powered by lasers for excitation, has synchrony optics to crosstalk suppression during simultaneous acquisition, and benefits from a microlens enhanced spinning disk confocal. This one will give you even 100,000 wells a day for a 4-color assay. Both systems share really the same camera type, the same objective lenses, the same options for wide field and confocal imaging, bright field and digital phase imaging, and they share the very same mechanics that enable this very fast image acquisition. They have 6-position objective turrets with options for temperature and CO2 control. We have the Harmony 4.5 software, which provides you access to the features we've just been talking about. all of this is basically part of PerkinElmer's integrated suite of high-content screening solutions, which should only include the detection instruments we were speaking about before, but also sample handling solutions, imaging plates, really advanced image analysis tools, either in Harmony or in our server-based Columbus suite of things. And then last but not least, with High Content Profiler, a dedicated tool for multi-parametric profiling and hit selection. All of that truly supported by our support teams globally to enable you higher productivity and hopefully faster and better decisions from your data. Thank you.
Thank you, Dr. Tesdorpf. Our next speaker is Mr. Joe Trask. Joe joined PerkinElmer in July 2016 and is instrumental in strategic customer support through teaching, training, and collaborations. He brings over 20 years of experience in high-content screening technology in academia at Duke University and Ohio State University, in the pharmaceutical industry at Abbott Laboratories and Eli Lilly and Company, and in biotechnology at the Hamner Institute for Health Sciences and Cytovation. Joe has extensive experience in cell-based technologies from flow cytometry, confocal microscopy, and computer-assisted automated microscopy studying cancer, immunology, neurodegeneration, and toxicology. Here you go, Joe.
Great. Thank you, Christina, for that. And I would, and thank you, Jacob, for the introduction. I think that's a great segue into my discussion today, which I'm going to talk to you about some of the applications that have been run on the Operetta CLS high-content imaging system. So just as a general impression for me, since I've just joined PerkinElmer in July of 2016, is that my impression of the system was fantastic. This is a great imaging system. I was really impressed with the high-powered LED light sources that Jacob was just mentioning about the hardware components of the system. So I'm not going to go through those, but I can tell you this is offers a great offering for the community. I'm really impressed with the Harmony image analysis software and some of the new tools that are available that Jacob just mentioned, especially the global image and PreciScan, which I'll be giving some more application examples of that. And I really want to just really start off and talk a little bit more about the 3D imaging aspect of the CLS instrument. So when we think about 3D imaging, and as Jacob pointed out, that these models are becoming more complex, and the reason for doing this is that we really want to try to recapitulate the in vivo environment. So when we challenge these cell models with compounds or drugs, we're trying to mimic that in vivo environment. And doing that, the 3D models really are a good way to do this in an in vitro plate. So when we start to look at these spheroids, for example, as a representation of a three-dimensional measurement, we'll capture these stacks, as we call them. So we get these individual planes that are shown here, and we can collect many of these at different z heights, as we call them, and these can be anywhere from 0.5 micron to several microns in size. So there's actually two ways to do three-dimensional measurements. We can either take the stack, or we can actually collapse. We can collapse the stack, and sorry here. And this will form a maximum projection intensity of all the planes together. And this is a way for us to do a very fast measurement. And this does not take long to do. So the maximum projection intensity collapse images are a much faster way to do analysis of three-dimensional image stacks. The other method is to do individual measurements on each plane. And when we do this in a relative analysis, this actually can increase the analysis time by several fold, depending on how many stacks you actually may have in the image. But the idea here is that we were able to, because the cells and the positioning of the cells are in and out of focus, collapsing them actually helps us project those into a plane that's easier for us to see and for us to analyze. So an example of this is recent data that our colleagues in Hamburg did for us, Alexander, and he used the Inspiro gravity trap ultra-low attachment plate, and this was a 96-well format. Approximately 2,500 HepG2 cells were seeded, and they were grown for 5 days. They were then challenged with a mitochondrial uncoupler compound called FCCP overnight, an 11-point dose response. Those cells were then labeled with a tetramethylrhodamine ester, and for about 45 minutes, and then washed, and then these were imaged on the Operetta CLS using a 10x objective. Reason we had to use a 10x 0.3 NA objective is because of the thickness of these particular plates. We did run it in confocal mode and also used the environmental control, which was 37 degrees and 5% CO2, and collected a Z-stack of approximately 7 planes, each being about 10 microns in size. And then a maximum projection collapse image was formed, and then we used the Harmony software for image analysis. So this is an example of some of the images that were captured from the 3D spheroids for HepG2 3D spheroids. And you can see a DMSO control, and as the intensity, when you have high intensity with tetramethylrhodamine, that is good functional activity of the mitochondria. But when FCCP, which is a mitochondrial uncoupling agent, disrupts the ATP synthesis, it will start to decrease the fluorescence. And you can see that occur as we increase the concentrations of the FCCP compound. And when the time we get to 300 and at 1,000, you can see that they're at 300 micromolar, there's hardly any visualization of the cells. They're basically broken up. The spheroid has broken up, and at 1,000 they have disappeared and likely have washed off the plate. So we were able to take that data and generate a dose response curve, and the fit went pretty well for this. Again, these were cells that were challenged just overnight, so approximately 20 hours. And you can see we have a nice dose response there where we can actually calculate an IC50 curve, and this was N of 3 different wells. So, and I'm not going to go back because Jakob gave a nice overview and introduction to the PreciScan, at least the principles of it. So, it's a method we know that we can do a pre-scan and of cells, and then go back and do a rescan at higher magnification, this is a technique that's really been needed in the community for a long time, and I'm really glad that PerkinElmer has brought this into their portfolio. It's really easy to use. It's all embedded into the Harmony image analysis workflow. It's very flexible. So essentially what we're doing, there are 2 things we have to do. We have to basically create a pre-scan algorithm, and then we also have to create a post-scan or rescan algorithm. So you're creating these 2 different algorithms to do the analysis. And so this is going to be very, very nice, and I'm going to use this example of the mitotic index identification using HeLa cells. HeLa cells, of course, many people know, are one of the oldest cell lines available from ATCC, and it's immortalized, derived from cervical cancer. Approximately 5,000 cells were seeded in a 384 well cell carrier plate and seeded overnight. And then they were challenged with docloxacin and with a dose response from 0.003 to 1.6 micromolar for 24 hours. And then the cells were fixed with paraformaldehyde and then labeled with 4 different probes. And so those probes were anti-phosphohistone H3 to measure mitochondria, phalloidin or F-actin, and they're labeled with TRITC reporter, and then used an anti-tubulin antibody labeled with Alexaflor 488, and then counterstained with Herx dye 33342. And so when we talk about what we're interested in this particular case of measuring, we're talking about the mitotic populations here. So I'm going to show you some examples of how we are able to measure a rare event measurement of mitotic cell, or close to rare event, and using a 5x objective lens, and this is using it in wide field mode. And then we switch it over to do the rescan at the different algorithm using a 63x water immersion objective, and then taking a small Z-stack of those other images. So this is just a screenshot, if you will, of the Harmony image analysis software, showing a 5x with all 4 channels shown here. And so that's the first step in the PreciScan method, is that we can identify these cells. And the next step, and we have the whole arsenal of the Harmony image analysis tools to make evaluations based on the intensity of the probes we're interested in, the size, the shape, the texture, and even dynamics of these individual objects. So now we can go in and identify these individual objects, shown here using segmentation routines, and then we can next identify a threshold, and the threshold is actually used to identify the positive cells, the cells that actually we want to go back and rescan. So you can see there's the red line that's shown here at the bottom of the screen, shown here. And that is our threshold point, and then we're going to actually identify the cells we want, and that's going to be shown here and here. So those are the positive cells we identify that are positive for phosphohistone H3. So these are the mitotic cells that are in the overall population. So we're looking at a subpopulation within the context of the cells that were in the well that were challenged with oclosamine. So here's a screenshot of the DMSO control showing you the number of objects that were actually identified. And so these are the scan areas at 63x. So these are the areas that the system's going to go back and rescan these particular areas to identify the mitotic cells. And each one of these boxes, and if you can't see a cell behind it, it's because the numbers are overlaying it. The precision on this is very accurate. So almost every cell, if there's only one cell in there, is dead center inside that new scan area that has been identified. So we increase the concentration of the compound, and you'll see that we'll start to have more and more. There's not much change here at low concentrations of the compound, but as we go into higher concentrations, you can see there's many more. So as the cells become arrested, we'll have more cells that accumulate into mitosis. So once we identify those, we can go back and then rescan them at 63x, and rather than showing you all those images on that single shot, I wanted to show you some of the phenotypes that were identified from this particular plate. So when we start to break down the mitotic phases, we can identify these based on some phenotype. So of course in interphase, we're talking about cycling cells here, so these are cells that are in G0, G1, S phase, and perhaps G2. And then we can identify cells that are in prophase, so these are phosphohistone H3 positive cells, so these are cells that are in mitosis in early stages of mitosis, and they're identified in the red arrows as shown. We move to there's 2 cells that are identified into metaphase, and so now we're starting to look at basically the band that goes across the nucleus, as shown here with the orange arrow, and then we identified 2 objects that appear to be in anaphase, and then in telophase and cytokinesis, we were able to identify 1 here. So we started to have the splitting of the mitotic spindles, and finally we identified 1, as you can see at the bottom screen with the green arrow. there were two identical daughter cells, we start to get that, the splitting and division of the cell. So the power of this new method of PreciScan, we're able to take a 5x image and now go back in the same well, in the same locations, and rescan and really get this detailed information. This is a power that's really been missing in the field. This is a great application, example to show. So as Yeka mentioned, global image is another method, and I want to talk to you a little bit more about using this particular technology, this application, to look at whole tissue imaging. So really approaching things that people may be doing in digital pathology world. So we can actually load up a slide. So this slide is it will hold this folder will hold 4 slides. It's SBS SLAS standard format, and this is just an example of the flow cells that were prepared, and to showing you images from using a 20x water immersion objective or even a 63x water immersion objective, and the quality that you can actually get using a slide holder. So the precision of the stage is very good, and actually the magnetic holders of this, of the slide holder, works very well for positioning these slides for scan and rescan. So example of this is a tissue section from a tonsil. So again, pre-scan, so we can measure, identify where the tonsil actually resides on the slide. So we can use that as shown here and here, and then we can identify the tissue regions. You're doing a rescan of the fields of view or the ROIs of interest. So just based on contrast, you can then identify the tissue itself, and then we can basically stitch these together or have a montage as a global image. But we could also go back and look at these individual fields in something we call a local image, so then we get the quantitative analysis that we need. So the composite images, there was 353 fields to capture this particular tissue. So it's a lot of scanning. So it went through and it scanned all of this. But we're not interested in all 353 fields on there. We're really interested in this particular case of looking at cytokeratin positive cells. So we masked using Harmony software, a region of interest for those. And as you can see at the bottom here on the that we have identified non-epithelial cells that seem to be a artifact that was present in the tissue sample, which is not uncommon for tissue preps. And we're able to identify the cytokeratin positive areas in here. So now we're able to just go back into those areas and rescan and look at subpopulations of analysis. So when we do that, we're able to again just look at a small field within that region. We call again local, and this local image we're able to identify the proteins and the targets of interest. And this is more telling when we start to look at, in this particular case, this tissue was stained with several different markers. The cytokeratin, the CD20 B cells, and CD4 cells. So we can start to look at the subpopulation of these immunological phenotypes. So again, here you can see within that cytokeratin positive cells, we can start looking at subpopulations within that layer. So we're talking about several different layers that we can start to look at and look at these subpopulations. Both at CD20 using B cells, and then also looking at CD4 T helper cells. And you can build this list on looking at other immunological phenotype measurements, because it's really going to depend on your question and what you're interested in looking at. This technology, this approach really opens up a lot of avenues and doors. It's something I wish I had several years ago. Thinking about inflammation from tumor invasion, that you have this immunological response, that you can start to look at cytokines, chemokines, and other phenotypes, other CD markers. including HLA, DR. So I think it opens up a lot of opportunities for the community. Also, and you have to think about this, is the only way to do this is with intact tissue. It really keeps everything in context to the environment. And the only way to do this before properly was to use flow cytometry. So they'd have to dissociate the tissue and then run it by flow cytometry. But then you lose everything in context. So now you can't measure where these particular phenotypes are in relationship to a tumor infiltration, for example. So I think that there's a lot of promise here, and it's going to be exciting to see how research blossoms from this technology, and looking forward to seeing that. So I'm going to just really close and just kind of mention some of these other cell models that you could think about using PreciScan and global image for, because really it's a method for pre-scanning and rescanning cells, tissues, and organelles and other things. And Jacob's already mentioned the scratch assay, and that's just an image from 4 different fields to show that. But this is work I had done when I was at Duke University with Don Lowe and measuring ex vivo brain slice imaging. So this is quite a large cross section of a live brain slice. So we can go in and use this type of method to pre-scan this and then rescan at higher magnifications. And then with global image, we can start to look at these dendrites and axons that are going across the particular tissue and measure that in context rather than having to break it up into separate fields. So I think that there's a very nice utility and power of having that. Another example is some work I had done in the past with Hepatopac, which is a co-culture model. So this is a micropattern of primary human hepatocytes on a monolayer of fibroblast cells. And now we can go in and identify these individual islands of hepatocytes and make quantitative analysis just on those, very much like the microtissue approach. And also organelles. You have to think about organelles. It can be C. elegans, but it could also, you can look at other organelles systems, including flies or even zebrafish. So I think there's a big promise of using this scan and rescan methodology. So with that, I'm going to stop there, and I want to thank you for your time.
Thank you, Joe. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, does PerkinElmer provide any solution for image storage?
Thank you, Chrissy. That's a question I'd like to take. This is Jakob Tesdorpf speaking, and thanks for listening in. Actually, yes, we do. Obviously, there's a little bit of question on that. Is it a hardware-related question? In that case, there are a lot of third-party suppliers for appropriate hardware. When it comes to software, we offer our Columbus image storage and analysis system to take care of this. So the answer is absolutely yes.
Thank you. Next question. What is the maximal tissue thickness that can be analyzed with the Operetta CLS?
Okay, I think that's a question I'd like to answer as well. This will depend to a great extent on the type of tissue you're analyzing. I assume this question relates to typical 3D spheroids or microtissues. We've done extensive work on this type of tissues with different instruments now. And basically, two things are key here. One is the water immersion objectives that really help you to get deep into the tissue without distorting the image. And the second thing is the design of the spinning disc. The Operetta CLS has a classical spinning disc design without microlenses and a pinhole spacing of about 250 micron. And in that case, we typically see a depth of up to 50, 60 micron, maybe sometimes a little bit more than that, where we can still successfully segment all the nuclei in a given plane. This depth can be improved upon by clearing the microtissues if it's an endpoint assay, in which case we typically can double the depth that you can still analyze, or if you have predominantly need for working with microtissues, you're also free to use an Opera Phoenix instead, which has a larger pinhole-to-pinhole distance, which also translates into a deeper depth of analysis in these tissues, which is typically also then in the range of roughly 100 micrometers for a typical type of tissue. If you're interested, I can certainly send some application notes down on that topic.
Thank you. Our next question is two parts. Now, you talked about HCS assays using 3D models. What are the biggest challenges in imaging and quantifying 3D cell models, and do you have any advice to overcome them?
Thanks, Christina. This is Joe Trask speaking, and I'll take that question on. I'd first like to say that at last week's SBI2 meeting, Society for Biomolecular Imaging and Informatics meeting in Boston, there was a 3D colloquium to really talk about some of the challenges with 3D imaging. Of course, as Jakob pointed out, light is the main thing. So getting good light penetration into the tissue so we can actually detect it, is a challenge. And so it really comes down to the size of the spheroids or the tissue that you may be looking at, and what how far light can penetrate into the tissue and then be detected. It also has to do with how those measurements are going to be made. So as I mentioned, in my slides, is that you can collect a series of Z-plane images and you collapse those into a maximum projection image. And that seems to be the way most people are treating 3D image analysis these days currently, although there are some groups that are actually looking at the volumetric, the voxel space. If the biology warrants it, they're going to need to do that if they need to look at particular objects in three-dimensional space. So it really depends on models. Now, ways to, advice to overcome these, it really comes down to your assay development. So you need to go through, those steps of when you develop your assay and validate it to make sure that, those are reproducible. And there are some groups that have found that having very large spheroids, in fact, is not a good thing. In fact, they prefer to have smaller spheroids for the purpose of light penetration, but also for dye. So getting the dyes and probes into the cells for labeling is very critical, so you can actually detect those in context of the tissue. So I think it really starts, again, about assay design and development and validation of the assay.
Thank you, Joe. Next question. Is there an option of having a color camera or a color filter on monochrome camera?
Yeah, let me take that one, Christy. So I'm not 100% clear to what imaging mode this question relates. So if the question relates to fluorescence imaging, then we obviously have emission filters in place to adjust the camera settings there and filter the emission light. When it comes to bright field imaging, in that case, the answer is we do have a single wavelength bright field source, which has been selected to really maintain cells healthy and alive and to minimize the light exposure of the cells, which is not well suited for colorimetric assays, for example, like H&E stains.
Dr. Tesdorpf, I believe you can answer this one as well. Can we do wide field, then switch to confocal?
Yes, absolutely. So this is perfectly possible in this system. We can offer both modes in the system.
Very good. Next question. For image analysis, do you have different modules to address different assays? say translocation, cell cycle, granule assay, nutrient outgrowth, etc.?
The answer to this is actually yes. Harmony software uses an approach to which we call the building block approach, which enables users to easily assemble specific protocols for specific assays. But it also comes with pre-assembled protocols that we call ready-made solutions, and they address common assays exactly like the ones mentioned here, like translocation, cell cycle, nutrient outgrowth, etc. They can be easily amended as well, so if the standard solution doesn't fit the assay of the customer and the question in hand, they can be easily edited by using the very same building block approach.
I believe we have time for one last question. Can you talk about the tools that the Operetta CLS has for phenotypic screening or phenotype classification?
While I'm at it, I might as well continue. We have a very broad range of parameters that we can read off samples using our Harmony software. Very powerful, specifically for phenotypic classification, are advanced parameters such as texture and what we call star morphology, because they have been specifically designed to answer this type of question. This allows the user to extract hundreds of features from each cell, and then we use supervised machine learning inside Harmony, a tool we call Phenologic, to identify the combination of the most relevant parameters to tell different phenotypes apart. If that is not sufficient for the question at hand, we can transfer data to another tool, which we call High Content Profiler. It's based on Spotfire, which also enables a completely unsupervised machine learning to do feature reduction and multi-parameter analysis.
I would like to once again thank Dr. Jacob Tesdorpf and Mr. Joe Trask for their presentation. I would also like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through March 8, 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see you next time. Goodbye.